Hello and welcome back. Today we are diving into cellular respiration and all of the parts that make up aerobic cellular respiration specifically. So this has a lot of chains and cycles and processes that unfortunately you are just going to have to sort of memorize. There's no super easy way to just sort of know this. There's a lot of information. I know there's a lot of information, but I think we'll be able to get through this. So let's get started. To begin with, one of the main pieces of cellular respiration is the production of ATP. So let's talk about what ATP is. It is energy, right? You've all heard that like the mitochondria is the powerhouse of the cell. Yeah, it's the powerhouse of the cell because it makes energy. That's why they call it that. It is literally the power source for all cells. It does everything that, like every movement that you do, that you think about, that you do unconsciously, like when you're sleeping, that you do unknowingly, like digesting food, all of that is run by ATP. So it does have 3 covalently bonded phosphates, right? So adenine is the A, and then triphosphate, 3 phosphates. That's what ATP stands for, adenine triphosphate. So 3 covalently bonded phosphates. When phosphorylation happens, it makes the molecules less stable, which is why ATP is super reactive, because it has really high energy bonds. And so, again, the functions of ATP, the key functions, is that it's the energy of the cell, and it will transfer its third phosphate group to other organic molecules, which makes them more reactive. So once you break this bond up here and you turn ATP into ADP, diphosphate, once you break that bond, you release that energy, and that phosphate can then be sent to other organic molecules, and you can use this energy that has been released. So ATP is synthesized from ADP, right? You have to be able to turn this diphosphate back into the triphosphate. It's synthesized back from 2 sources. If you photosynthesize, you can get this energy from solar energy, or if you go through cell respiration, which is what humans do, it's through oxidative processes, which we'll talk a little bit more about in the next slide. So, redox reactions. This is a throwback to chemistry. This is something that you have most likely learned already, but let's just do a quick review in case you don't remember what redox reactions are. So cellular respiration as a process involves the breakdown of organic compounds to produce ATP, right? We've been talking about ATP now for this entire time. When you stagger the breakdown, the energy requirements are reduced. So that means that you don't have to use as much energy as an input to get all of the energy that you typically would get as an output, because you're doing small breakdowns instead of one major huge breakdown. The released energy is not lost, so when you are doing this staggered breakdown, the energy is not lost, it's just transferred to specific carriers via these redox reactions. And redox is reduction and oxidation, right? So the RED of redox is reduction, and then the OX is oxidation. It's just an abbreviation. So reduction is a gain of electrons, a loss of oxygen, and oxidation is the loss of electrons and a gain of oxygen. So you can kind of see this here. This large activation energy would be too much energy for cellular respiration to be using continuously. It would just burn through all of our energy reserves to have to use this huge input just to get this like loss of energy as heat. And it's just like, okay, that just seems silly. Whereas these small activation energies, look at how much less energy is used and lost as you go through. Yes, there's a lot more steps to it, but you don't lose quite as much as the first picture. So I mentioned carrier molecules on the previous slide, and they're called hydrogen or electron carriers because they gain protons or electrons. We are going to talk about two really common ones. You're going to see them throughout the entire process of cellular respiration. They're NAD+, which is reduced to form NADH, right? You can see here we have our hydrogens and our electrons in this formula. And then less common but still present in cellular respiration is FAD, which is reduced to form FADH. So again, all of these are just carrier molecules to bring electron carriers throughout the entire process. So these hydrogen carriers are like taxis. They just transport electrons to the cristae of the mitochondria, which is just inside the mitochondria. But you can see it here, like you have your organic molecule, it gets broken down, and you put those electrons and hydrogen ions into your NAD plus or your FAD, and then you gain that energy and it becomes NADH plus a hydrogen ion, okay? So it helps move things along through this process. Now our first step of cellular respiration happens with or without oxygen. It's pretty impressive. Glycolysis happens all the time, and it happens in the cytosol of the cell, and basically that's just the cytoplasm. So it doesn't actually happen in the mitochondria, but it is the first step of cellular respiration, and the rest of cellular respiration does happen in the mitochondria. So this is like a little bit of an exception to the rule, but still very, very important. So there are four main events that happen in glycolysis. There's phosphorylation, lysis, oxidation, and then ATP formation. So phosphorylation is the idea that glucose gets phosphorylated by two ATP molecules. So an input into this to start glycolysis is two ATP molecules. You need energy to start anything. You can't just magically make something happen. You need to have like a kickstart to get it started. So glucose is phosphorylated, which makes that molecule less stable and more reactive. So you're kind of like putting a lot of pressure on glucose because you don't want a 6-carbon sugar, right? Glucose is a 6-carbon sugar. We don't want that. That's too big to work with. So once the phosphorylation happens, the next step is lysis, where glucose is then split because of all that pressure and the reactivity that we had done in step 1. In step 2, it is split into 2 triose phosphates, meaning 3 carbon sugars. So we went from a 6-carbon sugar, glucose, to 2 3-carbon sugars. We just broke it down in half. Then step 3 is oxidation. So we are removing hydrogen atoms from those 2 3-carbon sugars, and we reduce NAD plus to NADH, right? That's those taxis that we had just talked about. We are just loading up the taxis. With this, because we're removing hydrogen atoms from each of the 3-carbon sugars, we are making 2 NADH molecules total, right? 1 from each of the 3-carbon sugars. And then finally, ATP formation. Some of the energy released is used to directly synthesize ATP. Okay, this is just like, yay, we did a little bit, we got a little bit of energy back. With each of the 3-carbon sugars, we have 4 molecules of ATP generated. So with that, that's great, but we used 2 to start. And so because we used 2, we only have a net gain of 2 molecules of ATP, which you can see here. So this, again, is just a breakdown of exactly what I was talking about. So we start with glucose. We use ATP to put that pressure on glucose to make it unstable, and when it becomes unstable, it will break down into 2 3-carbon sugars. From here, hydrogen ions are released from these 2 3-carbon sugars, and we have our first kind of introduction to our hydrogen carriers, our little taxis, and from here the NAD plus takes on the hydrogen and becomes NADH and an extra hydrogen ion, okay? You see that happening. And then through the rest of it, we do generate 2 ATPs per 3 carbon sugar. So we generate a total of 4 ATPs in here. But again, we needed 2 to start this process, so the net gain is actually only 2. So here at the bottom, our net gain is 2 ATPs and 2 NADHs. And our final product is 2 pyruvates. That'll be important for our next step. But before we go on to our next step, the choice is up to whether there's oxygen or not. So there are 2 paths you can take after glycolysis. You can go the anaerobic way or you can go the aerobic way. So anaerobic is if there's oxygen not present. So if oxygen isn't present, then the pyruvate that you just formed in glycolysis is not broken down further and you don't get any more ATP. That is not a lot of energy being produced, so it's not ideal, especially for a human. You need lots and lots of ATP, and so only getting 2 is like, okay, now what? And so that pyruvate remains in the cytosol and is then converted, depending on what organism is doing this, into lactic acid. If you're a human or you're bacteria, it will be converted into lactic acid, or ethanol and carbon dioxide if you are a yeast. This process, though, is reversible. So once oxygen does become present, then that lactic acid and ethanol and all of that can go back, and the pyruvate can then continue on into our next step, the link reaction. So although anaerobic is not ideal, there are times, even as a human, when you are running low on oxygen, like when you are exercising too much and all of a sudden you have that huge cramp in your side. That's not just a cramp, that's a buildup of lactic acid because you have run out of oxygen there. Now on the flip side, what you're typically used to is aerobic respiration, and this is when oxygen is present. So the pyruvate that was formed at the end of glycolysis is transported to the mitochondria to get broken down even further. This will generate a huge number of hydrogen carriers, and with that, you can make more ATP. And that's like, we like that. We want ATP, we want energy, we want to be able to do stuff in our lives, right? So ATP, vital for just like living. So let's assume that we have oxygen. We're going to dive into aerobic respiration. So with aerobic respiration, our first stage is the link reaction. So this is taking the pyruvate that was formed via glycolysis, and we're going to transport that into the mitochondria now. Remember that glycolysis happened in the cytoplasm, so now we are actually in the mitochondria. So this reaction links the products of glycolysis with the aerobic processes of mitochondria. That's why it's called the link reaction, because it links the 2 pieces together so that you can continue on. So the pyruvate is transported into the mitochondrial matrix. The pyruvate loses a carbon atom, so you have CO2 forming, and the 2C compound, right? Because when you have pyruvate, it's a 3C compound, right? Glucose was 6C. It got broken down into pyruvate, 2 pyruvates, 3C each, and then we just lost a carbon to form CO2, so now we're down to 2 carbons. Okay, so that 2 carbon compound forms an acetyl group where it loses a hydrogen atom, okay? And so when that hydrogen atom gets lost, it takes NAD plus and turns it into NADH, okay? Our taxi is back, picking up another passenger. The acetyl group combines with coenzyme A to form acetyl coenzyme A. This is just a compound, 2C compound that you should get familiar with. This is gonna pop up from like time and time again. It's a fairly common molecule that we talk about in science quite a bit. So per glucose, remember glucose is a 6C, so per that you're doing this link reaction twice, right? Because the 6C glucose gets broken into two 3C pyruvates. So this picture that you are seeing here happens twice. The pyruvate, this is half of your glycolysis outcome. So this will happen 2 times. So you will have one pyruvate, lose a CO2, lose a hydrogen ion, your taxi will come pick it up, and then coenzyme A will join up and you will create acetyl coenzyme A. So per glucose molecule, you will end up with 2 carbon dioxides and 2 NADHs. And 2 acetyl coenzyme A's, of course, because that's your outcome. Okay, the second stage of aerobic respiration, this occurs in the matrix of the mitochondria. So now we're starting with the end result of the link reaction. So our starting point is acetyl coenzyme A. And acetyl coenzyme A transfers its acetyl group to a 4C compound called oxaloacetate. Okay, I know, there's just like so much, and you're like, are you serious? Why do we have to do all of this? It's just a very, very important process, and you just need to like remember these terms. I know, but the key here is acetyl coenzyme A is 2C. Okay, 2 carbons. It transfers its acetyl group to a 4C compound. I'm like kind of less concerned that you know the exact terms and you understand that 2C from acetyl coenzyme A, 2 carbons, plus the 4 carbons from oxaloacetate makes a 6 carbon compound, right? 2 plus 4 equals 6. That just is like, that's math. That's how that works. And so citrate is the key here, okay? So when you start the Krebs cycle, you are starting with a 6 carbon compound. And then a series of reactions occur to break the 6 carbon compound down to reform that original 4C compound. And you're probably sitting there thinking, so I have to know this whole Krebs cycle just to know that I built something to immediately break it down? Yeah, you do. Yep. Yeah, you do. And that is because, remember, we talked about doing multiple mini steps to ease the loss of energy. And that's exactly what this Krebs cycle does. It does a lot of small steps to break things down, to get a lot of items like those electron carriers, those taxis. And we do this because it's easier on the energy output. So as you're going through these reactions to break down the 6C compound, you are getting 2 carbon atoms released, and that forms CO2. Lots of oxidation reactions, which is results in a reduction of hydrogen carriers. You get 1 ATP molecule. Remember, you have to do this twice. And these series of reactions turn citrate back into oxaloacetate. So it turns the 6C back into the 4C. So remember, I said it, but I'm going to say it again. This occurs twice because the link reaction produces 2 acetyl coenzyme A molecules. So each of the acetyl coenzyme A molecules goes through this Krebs cycle and produces this net outcome. Okay, so let's take a peek at what this looks like. So again, you have acetyl coenzyme A as your starting point. It is going to combine with oxaloacetate to form citrate, this 6 carbon molecule. Once that 6 carbon molecule is there, you're going to start going through a number of steps, right? Right off the bat, we lose a carbon to make CO2. Then we lose a hydrogen where NAD plus becomes NADH. Again, our taxi is picking up more passengers. So now we're down to a 5C carbon because we lost one for carbon dioxide. In our next step, taxi's picking up another passenger and we lose another carbon to CO2. Now we are a 4C molecule. We also got a little ATP, a tiny, tiny, tiny bit. 1, to be specific, 1 ATP, a very tiny bit. And then as we continue on from our 4C, as we're finishing up the Krebs cycle, our taxis come back and pick up some more people, but this time it's not just NAD+, we are also seeing FAD because there's so many passengers to pick up here, we needed 2 taxis to come and grab them. So remember, this happens twice per glucose molecule. So our net outcome per glucose molecule, which is how you should always think of this, is 4 carbon dioxides, 2 ATPs, 2 FADH2s, and 6 NADHs. Okay? So we're not doing a ton of stuff here. We're not like really changing anything. We're just breaking things down in small steps so that the energy use is minimal. All right, we're almost done. We only have one more step in aerobic respiration. The final stage. Da-da-na-na-na-na. We did it. We almost made it. Except this is the most complicated. So this occurs in the inner mitochondrial membrane, aka the cristae. It is folded up. The cristae is like lots of tight little folds for additional surface area. The more surface area you have, the more electron transport chains you can be running at the same time, which means the more ATP you can be making. So the ETC, electron transport chain, I will abbreviate it to ETC from here on out. The ETC is broken down into multiple steps. Step 1 is generating a proton motive force. And basically what this means is we need to set this whole process in motion. And the way that you do that is you take all of those NADHs and those FADH2s that were oxidized and release high energy electrons and protons. So you bring all of those taxis that you have been picking up passengers with throughout this whole process, and you bring them to the ETC and you say, all right, get out of my cab, right? You let those hydrogen ions and you let those electrons out, and the electrons are transferred to the ETC. And you can see that that's this yellow line here, right? The electrons are transferred there. The electrons pass through the chain and lose energy as they pass through the chain. That energy is used by the chain to pump those hydrogen ions, those protons, from this matrix. You are going to accumulate a lot of hydrogen ions in the intermembrane space, and that will create a gradient, right? When you have too many hydrogen ions on one side, that's high concentration, and not enough on the other side, that's a low concentration. That's just a gradient in like general terms. When you have this accumulation of hydrogen ions, you are creating the proton motive force. Okay, you want to have an uneven balance of hydrogen ions, because when you have that uneven balance, that's how you can create this force. Step 2 is ATP synthesis via chemiosmosis. So that proton motive force causes hydrogen ions to move down the gradient and diffuse back into the membrane. So we are pushing hydrogen ions through all of these compounds, and that's great. We're using that force, but the problem here is your body likes to be in homeostasis. It does not like to have an uneven, balance of these ions. So we're going to start sending the hydrogen ions back. So the diffusion is called chemiosmosis. It's facilitated by the transmembrane enzyme ATP synthase. So when the proton force causes the hydrogen ions to move down the gradient, we want to diffuse them back into the matrix. And by doing that, that process of diffusing the hydrogen ions back is called chemiosmosis. And the way that it is able to diffuse back is through this green enzyme at the end that's called ATP synthase. As the hydrogen ions move through ATP synthase, it rotates. The enzyme, the ATP synthase, rotates and synthesizes the ATP as it's rotating. So it's actually pretty cool. It's like a weird trick that it does. You don't really see a lot of enzymes rotating, but this one does. So cool. And then the final step here is the reduction of oxygen. So in order for the ETC to continue, those de-energized electrons have to be removed. Remember, we sent the electrons through all of these compounds to give energy. Well, now they're dead. Well, they're not, well, you know what I mean. They're like a dead battery. They have no energy left. And so we don't love that. We need them to be energized. And so oxygen acts as the final electron acceptor. So this oxygen right here that you see will pick up this de-energized electron. It will also bind with the hydrogen ions that are being diffused through ATP synthase, and it will form water. When it forms water, it helps to maintain the gradient so that it doesn't get out of whack and it doesn't sort of mess with the process that the ETC will typically go through. If oxygen is not present, you cannot produce ATP. It is absolutely vital that oxygen is there to pick up these extra pieces, this de-energized electron and these hydrogen ions. If oxygen is not there to do that, the whole process is halted. And if you can't make ATP, why are you doing this, right? Because that's all we care about, really, is the production of ATP. So we talked about this a little bit. There are 3 types of aerobic reactions: decarboxylation, oxidation, and phosphorylation. This is just a nice little chart that shows when these things happen. So decarboxylation is when you are losing a carbon, right? So we lose carbons in the link reaction and the Krebs cycle. Oxidation is when you are using your taxi to pick up hydrogen ions, and we see that in almost all of the steps except the ETC. And then phosphorylation is the production of ATP, and that happens in all steps except the link reaction. It happens the best in the ETC. That's why the ETC is so important, because it makes 32 ATP, which is huge. So this is just a summary. This is a nice image that shows like net gains as you go through the entire process of aerobic respiration. I'm not going to go through it because I just spent like 30 minutes going through it, but this is just a nice picture. And this shows the production of ATP and how it all happened. So again, fantastic. It's just another image to show exactly what we were just talking about, but in a different way. The mitochondrial structure, you should be able to both draw and understand this. So this is a mitochondria. You have inner membrane space. You have the matrix, which is the yellow part. You have the outer membrane, which is like this dark red. The cristae are the folds inside, and then the inner membrane itself is where the electron transport chain exists on the membrane, so on this like light pink inner line. Okay, so you should be able to draw this and label it exactly as this picture shows. So very quickly, electron tomography is used to model a 3D internal structure. So basically you can use electron tomography to model what the mitochondria looks like as a 3D structure. So samples are imaged using a transmission electron microscope. It's tilted to get a different angle after every single image, and then compiled to reconstruct a 3D model, which is the tomogram. And then they're usually, the samples that you're using are usually fixed, dehydrated, or frozen so that you like have them. So with this process, we have been able to find a number of features in the mitochondria. So we now know that the cristae are continuous within the internal mitochondrial membrane. That's cool. The intermembrane space is a consistent width throughout, so that it doesn't get bigger or smaller, it just stays the same width. And now we know the relative shape, position, and volume of the cristae can change in active mitochondria. So although they are continuous, where they are can change depending on what the mitochondria is doing. Pretty interesting. And then our last two slides are going to talk about the phosphagen system, which is an energy storing compound mostly found in muscle and nervous tissues. This is immediate access to the reserve of high energy phosphates to make ATP. A common phosphagen used by animals is phosphocreatine. So when you are at rest, ATP is hydrolyzed to ADP, and the phosphate is transferred to creatine. Okay, you see that here, right? That phosphate is transferred to creatine, and then when you're doing exercise, the phosphocreatine is hydrolyzed, and the phosphate is released to then make ATP to be able to continue doing work. Okay, so this is just like, this is how we keep our high reserve so that we don't immediately run out of ATP the second we like wake up from sleeping. And then energy systems. So this is our last thing. This is just showing you like how your energy is able to last or not last. So phosphocreatine synthesizes ATP rapidly, but the reserves are basically gone in like 30 seconds. So it's great, but you know, it's not the best. Anaerobic respiration provides a little bit better of a pool of ATP, but it will eventually produce lactic acid, which is not ideal, right? And then aerobic respiration requires oxygen, of course, but because it produces such a large amount of ATP, it's kind of like a better system. It's not kind of, it is the best system. And so you can see here in green, the phosphagen system is like great for like sprinters because you get all that energy right away, but then it's like, whoop, I got no energy coming out as we continue on. Anaerobic is we have a nice little bump, but then it goes back down because we're making lactic acid at this point. And then aerobic is like, yes, we love that. It's a slow start, but you can maintain a higher amount of energy contribution, so we like that because we're able to continue on for a longer period of time. Which allows us to like actually do stuff. All right, I know that was wild, that was crazy, that was like way too much information, but we're gonna practice so much in class that I feel confident that you will feel better about this as we continue on. So great job, I'm proud of you. I know this was a lot to write. I'll see you in class.